So now we are going to continue with the session of questions that we already have from our students from biology and anatomy and physiology from Hostos. So we are going to have sorry about that. Let me see. So we're gonna have the first session of the questions, and you will be able to answer these questions as we speak. The first questions are going to be introduced by one of our students. So Maria, please, can you go ahead and you can introduce this set of questions, please?
Yes. Hi everyone. These questions were asked by Dariani Munoz, and she wanted to know, what should I do if I had contact with someone with coronavirus? If I have symptoms of COVID-19, what should I do? And I took a Theraflu flu when I had seasonal flu. Can I take similar medicine to protect myself from COVID-19?
Thank you very much. Any of you want to address this question? You can both, Maite or Dr. Sánchez or Dr. Diago.
we can share, so we can split them. We cannot hear you. The microphone, Ellie.
Sorry. Yeah. Now.
Yes. Perfect.
Sorry. So I think if you want, I can answer the first two with the recommendations of the department, and the later is for you.
Yeah. Okay.
So if you have contact with someone with coronavirus, you should avoid as much as possible all contact with other people. So if you are not showing symptoms, you should wait. I mean, because there is nothing that you can do in order to protect that you or prevent that you later develop the disease. But you should avoid as much as possible contact with other people, stay at home, monitor yourself for symptoms. And if you have to leave home, again, wear the mask in order that if you ever become infective, you prevent the disease to be transmitted to other people. And if you ever develop symptoms, you have to for sure stay at home. And if those symptoms doesn't go worse, the disease is manageable with over-the-counter medicines, and you can contact to the 311 phone of the department to ask any questions. And the guidelines right now is if you show symptoms, you have to wait at least minimum 7 days to go outside of your home, or 3 days after your fever is resolved without any over-the-counter medicine. So minimum 7 days at home with, or 3 days after the fever is resolved with medicine, whichever is longer. So if your fever lasts 5 days, you have to wait at least 8 days to leave your home. So this is what is recommended right now. But if you ever have questions, the webpage of the New York City Department of Health includes all this information, and you always can call the 311 phone in order to ask your questions, and they will direct you to a medical provider.
I have a question regarding to this. Even if the symptoms are severe, even if the symptoms are really, really strong, you have to wait.
No. If your symptoms are severe, you should contact your normal healthcare provider in order that they tell you what to do, but you shouldn't be waiting. If you think that you have shortness of breath, okay, and you feel worse, you have contact your healthcare provider. What I said is if your symptoms are manageable with over-the-counter medicine. So it's like a cold.
Mild symptoms, mild. Yeah. And also if you don't have a condition, I believe, if you have asthma or if you have respiratory conditions, I believe that you have to go to the hospital?
Not go to the hospital. I recommend you to contact your healthcare provider or call 311, that they are going to direct your call to a healthcare provider to see what is best for you. It's better not to go to the emergency care showing symptoms without them to be aware that you are going to the hospital, in order that you prevent the spread in the healthcare system.
Okay, and you don't get infected there either, I believe.
Yeah.
Because that's also a place that you can get infected. Thank you very much.
Thank
Maite, would you like to go ahead with the...
Sure. Okay, if I took Theraflu when I had seasonal flu, can I take a similar medicine to protect myself from COVID-19? That I will leave it to the doctors that are the ones who know what you can take or you cannot take. So far, similar medicine to Theraflu is being under development to treat the COVID-19. There is some drugs that they see that are working better than others, but everything is still under research, and we don't have yet a specific drug to treat the COVID-19.
Okay, thank you. Let's continue to the following question. Maria Fer?
These are questions from Afnan Faria. Does COVID-19 infect children, and do the stools transmit the COVID-19 to the mother when she is cleaning the child?
Me? Yeah, so it infects children, although luckily it's not like the flu virus, because luckily the children, the symptoms that they are showing are much milder than the people with older ages. So it's true that they get infected, but usually the disease goes mild with them, or even they are asymptomatic. And the problem with that is that being asymptomatic, that doesn't stop them to transmit the virus to other people. So that's why children should be kept from being close to people of high risk, because even if they are not showing symptoms, they could be infecting other one.
Great.
Yeah.
There were also like many cases where like many kids have also like passed away because of the virus.
Give me 1 second, I'm gonna pin you. Sadia, can you repeat your question?
Oh yeah, sure. So I was actually like looking at the news, and like there are also like many babies who also like passed away from the virus.
Wow.
The percentage is very low. Of course, this is in a population, you will always have people that die because of, I mean, it's like the probability is, it is not zero, but it's true that the probability of getting a severe disease in children is much small than in an old person.
Right.
But it's not zero. That's for sure. Yeah.
Great. So,
Okay.
Maite, would you like to add something to this?
No, it's what the list says is what is happening.
Perfect. We're going to do the following questions. I think it's one of the last questions on infection and symptoms. I organized them in this way, so it would be easier. Maria, can you go ahead, please?
Yes. The first one is my personal question is the virus changing genetically as it continues to spread? Then we have a question from Elizabeth Mota, who says, how fast can a person catch COVID-19? And lastly, Alondra Ramos says, how does COVID-19 differ from other viruses, and what makes COVID-19 more dangerous?
So I will give the word now to Maite or Elizabeth.
I think Maite would be better.
Okay, I can answer the first. So the first question, is the virus changing genetically as it continues to spread? The answer is yes. This virus has a mutation rate, which is not as high as other viruses, like for example influenza. It mutates in a lower rate, but yes, it still can change. So since it's a new virus, we don't know what is going to happen.
Will that make it more difficult for us to find a vaccine?
No, because I think the vaccine is gonna target specific genes or proteins that doesn't have this high mutation rate. So we will hopefully cover even if the virus changes.
Okay, thank you.
And regarding to the second question of how fast a person can take the COVID-19, I have a question as you were giving the presentation, Maite, that you were presenting this curve with the different tests at the different and the different states of this disease. So from which period of how long is this person able to infect other people? Because that is something that it would help us to understand how long do we need to be in quarantine to be to not spread the virus to our family members if we catch it.
So that's a very good question, and it's a key question that I think everybody is still investigating. We don't know. Some people can spread the virus from day 0 of infection to 3 weeks, 4 weeks. Now I think the media, the average is like from the day of infection until 4 weeks. So it's a long time. That's why we need to be in quarantine and stop. And do social distance.
I see. Yeah. And if you would like to address the last question, maybe.
Also, what I want just to add that how fast the person can catch COVID-19, it would depend on whatever I have said. If you stay very close to someone, is going to be, you get more chances to get transmitted the disease from that contagious person. If you stay for a long period of time, of course you will have much more probabilities to catch the disease, and so that is going to increase the chances that you get the disease.
Yes.
Mm-hmm. Let's continue. I think we are going to run a little bit later, but I think that these questions are very, very important for us to solve. Maria, do you want to go ahead and ask the last few questions, please?
Yes. Is how can we get ourselves protected from this virus? Is it safe to work somewhere while wearing surgical masks and gloves only? Are these 2 things enough to keep safe? Does coronavirus live in the air or on the surface? And does the high temperature end coronavirus?
Mm-hmm. We have very good questions from our students, as you can see.
Yes.
So who would like to start with that?
So maybe yeah, so how we can protect ourselves is washing our hands. That's the best thing that we can do in order to protect ourselves, and avoid to touch the face, like the eyes, the nose, because those are the places where the virus is going to enter. And if we have our hands contaminated with the virus, this is going to enter in our mucosa, and we are going to get infected. Also, avoiding being more than 6 feet apart of other people that we don't know if they have the virus or not. So social distancing, the hygiene, and wearing. So according with the going with the second question, the masks are not going to protect ourselves, because the type of masks that we are going to use are not those used by the healthcare providers, that those are also protecting them to get infected. So masks are going to prevent ourselves, that if we are asymptomatic and we are infected and we are shedding the virus without knowing, we will prevent others to get those droplets, or the droplets that we are shedding to be landing on surfaces that other people are later going to touch. So the mask,
yes, it's avoiding the spread. It's avoiding the spread, but it's not 100%.
No, for us, it could prevent something, but the most important thing is to don't touch our face and avoid the physical distancing and wash our hands continuously if we are outside and we are touching things that we don't know who has touched before. And the gloves, they give a sense of protection that is not real. The virus is not going to enter in your body through your hands. The virus is going to enter through the nose, the eyes, the mouth. So even if you are wearing gloves and you touch your face after touching something, that the contamination could occur. So we shouldn't be laying into wearing gloves and masks, thinking that this is going to protect ourselves. So no, they are not going to do it. So in my case, I don't use gloves because they are not doing anything. I mean, what I do is going with my gel, the alcohol gel all the time, washing my hands when I are outside and I have to touch things.
And do not touch your face when you are outside,
Yes.
right?
Yeah, and avoiding the distance, being 6 feet apart of other people.
Maite, would you like to add something to that?
No, I agree, totally agree with Ellie. And even if you wear gloves and you take the cell phone and then you start calling someone, forget about it. So it's better to avoid all the kind of touching and clean also your cell phones because we use them a lot.
Yes,
Yes, that's true.
Okay.
That's true. So regarding the healthy measures that we take, also I would like to ask regarding how long does the coronavirus live on the surface? So I don't know, do you have any idea?
Yeah, there has been research done on this, and some researchers has tested different kind of surfaces like wood, metal, crystal, glass, and it depends on the kind of material. But usually it's from 2 days to 3, 4, 5 days, things like that. And so in the surfaces, in the air, the virus doesn't live in the air. It only gets trapped in your droplets, in your saliva droplets.
Okay.
So it's not there. It's not a live entity. So it needs to be transported in the saliva.
I see. And the high temperature will end?
Yes, it will kill it, but it has to be super high temperatures, so like boiling temperatures or things like that.
Because they were saying that in the springtime,
in the summer, yeah,
it will be over.
Yeah. That's not high enough. Unfortunately, we see that that's not high enough. We were hoping to stop the spread in summer, but because of other countries that are now in summer and they still are having cases, we think the high temperatures are not gonna help us.
So basically, I think that you already answered the following questions regarding the food. So the virus is not gonna enter through the in our food, right? Because we have. I just wanna move a little bit to the following part. Basically, based on what we understood here, our students are concerned about the food. If the food can, if consuming food can put them on risk, or if the food, the virus can spread even in the refrigerator.
What I will do will be to wash everything, if it's possible.
Okay, okay.
Yes.
So you recommend to wash
and cook. If you cook something that is the high temperature is going to kill. Yeah.
Okay, so when you cook something, it's going to be the virus is going to go away, basically, right? And the windows, shouldn't we open the windows?
It's fine.
It's fine, right? Because
actually, it's good to open
the air.
Yeah, ventilation is what you need. If you are in a closer space with more people, what you need is ventilation.
Wonderful. Let me go to the next one. Regarding to immunity, Jasminda, would you like to ask that question? Or I don't know if she can, or Alex Faqir Afsalara. So he's going to ask the following questions. Alex, are you there?
Yes, Professor. Hi, everyone.
Hi.
So
Hi.
this question says, Can the virus COVID-19 become dormant in the cured patient and then became active to spread again? It became from Jasmine Otero. And the second question, it became from Eleha Azhar. She says, is that person affected by COVID-19 likely to get affected twice? And lastly, can people get reflected?
If reinfected, right? Basically, yes. Can we get the disease after we pass the disease, or we are protected after that? Or so basically that's the main, well, that's very important concern regarding the immunity.
I think it's under research still, because we don't know we are not completely sure that a person, the immunity that you develop after being infected prevents you to get reinfected again. But I think it's under research. We are still don't know. So yeah, there are cases that they were thinking that they were cured because they didn't detect the RNA after more than a month, and later suddenly they have the RNA, so they are not sure if they didn't clear the disease or they got reinfected. So it's something that is under research. I don't know, Maite, if you want to add something.
No, it's true. It's under research because it's a new virus and we don't know too much. The problem also is that all these tests and kits are also have, they are not 100, we cannot trust them 100%. So we can have false negatives or false positives, and that makes everything more difficult to address.
Regarding to this, it came to my mind a question that I had. So we have the detection of the IgM and the IgG, but we also have the testing methods of the PCR. And I wonder what is the fragment of the virus that is being detected on this PCR?
So it depends on the country. But most of them are detecting the N, the nucleoprotein, and one of the fragments of the spike protein. But there is a lot of variation among kids, but usually are these.
Okay. We're gonna go to the next question regarding treatment and vaccines. Please, Alex, do you wanna go ahead with these questions?
Yeah, sure. This question says, what is the convalescence plasma and why it is being investigated to treat COVID-19? And the second one says, what the treatments are being used against COVID-19, which of these treatments are showing positive results and why?
We already addressed a little bit of, about treatments, but based on your experience, what do you have more, I don't know, what do you think that is going to be the one that will let us continue to with our lives in the previous way?
Everything is so new that we are still doing research. So the one that Fauci talked about yesterday is giving good results and is going to clinical trials, and is, I mean, not to clinical, well, yes.
Yeah, it's into clinical trials, yes. Yeah.
And the convalescent plasma?
The convalescent plasma is also a very good one.
So in that I can talk a little bit. Oh.
Tell me, yes, go ahead.
Oh yeah, during my postdoc, what I studied was something about the convalescent plasma, because I was developing antibodies against, in this case, antibiotic resistant bacteria. So what it means is that when you are infected, you develop antibodies, and you expect that if you have recovered, those antibodies that you are developing are neutralizing the bacteria or in this case, the virus. So the convalescent plasma is getting the blood and getting the plasma from patients that have recovered from the infection. That plasma would be enriched on those antibodies that hopefully would have been able to clear the disease on those people that they recovered. So what you are doing is passively by injecting those antibodies to people that are suffering the disease at the, in another moment. So the clinical trials that are in place with convalescent plasma are two types. The first one is to prevent, to see if the convalescent plasma is able to prevent the infection of healthcare workers that are at high risk of contracting this disease, and to be able to help severe disease on people that are having this severe disease if they cure, or they help the immune system of those people to cure the disease. So, but this is something that they are studying, and still there is nothing. The problem with the convalescent plasma is that not everybody is developing the same antibody response, as Maite has said and has shown with the graphics. So maybe I cure the disease, I recover, but my antibodies are not the same as another person. So comparing different convalescent plasma from different patients that get cured is quite difficult. So that's something. So that's why this is the first thing that you do, but later to have something that is reproducible, as we want in science, is you get antibodies, monoclonal antibodies. So there is only one type of antibody that you treat patients with, and you see the same thing. You have to do clinical trials.
Because you can basically produce more harm to the people that if you inject some antibody from other person, no? You can
No, not for that. The problem is that you don't know the antibodies. Your response, maybe I cure, but what it helped me cure is not the same antibodies that another person produced. So in those clinical trials, it's difficult to compare. So that's why what you prefer is to isolate one of those antibodies that someone had and it is positive to neutralize the virus, and use that antibody for everyone in a clinical trial. So it's something more controlled. So the convalescent plasma, you do it because right now we don't have time to wait for getting a monoclonal antibody, because that will take you quite a lot of time. But I know that there are people that are working already in the research of monoclonal antibodies or partial FC or so.
Nice. So basically this question that we are addressing right now is related to the following one also. I'm gonna ask that right away. Why is it taking so long to develop a vaccine? That's a question from María.
Well, it's not taking so long. It's the vaccines in general always take very long, because you first need to develop it, have a candidate, and then you need to try it first in animals and then in humans. And you need to make sure that this new vaccine is gonna be safe, is gonna protect ourselves, and it's not gonna have any secondary effect that is gonna be worse than even the disease. So that's why it takes so long, because there is a lot of regulation and there are all of these developments which are necessary, right? Because we need a safe vaccine. That's why.
But even I would add that it's going faster than usual.
Yes.
I mean, in 6 weeks, Moderna put the first clinical trial with an RNA vaccine in 6 weeks. That's something that it was, I think, 4 weeks since they got the sequence of the virus until they went to clinical.
Yes.
No, definitely.
At all in any other disease, I think.
We are having some trouble with this microphone with Ellie.
Yeah.
So I just want to add related
working in the same.
Yes.
From everywhere. So I think this is so we have to be thinking. Oh, sorry.
No, you get a little bit cut, but you get a little.
Yeah. So what I was saying. Yeah. So what I was saying is that this this pandemic, what has brought is a huge international scientific collaboration that is speeding up the results, and everybody is collaborating as I have never seen in my career. And so I think this is something good, and we should be thinking like scientific community is doing everything as possible to get this disease under control. So I think this is, but you cannot speed up certain things because we need to do the scientific method. We have to go for the facts. So we have to get the results before doing any claims or anything. So this is what science has to do. Yeah.
Of course. So I wanted to ask, how do you feel? No, I think that you are kind of mentioning that it's like, we as scientists, we are always working, and people sometimes they ask, why do you do that? No, now it's a moment that you as scientists are working in the front lines. We can say it's in a moment that every hour of your work is valuable, and is everyone appreciate that. How do you feel to work? Is this like more exciting than usual that to go to the lab? How do you feel about that? I don't know.
Well, it's exciting and it's also scary, no? Because at the end, you do it because you like what you do, and it's a good opportunity to help more. But it's also is scary because we don't have the answers yet to everything.
Mm-hmm.
But as Ellie said before, the scientific community is really collaborating around the world. We have collaboration with Germany, France, US, many countries, and everybody is willing to help and to share. And that's the important thing, to share all the information, and in this way we can speed up what we can get, no, vaccines or antivirals or whatever.
Wow, that's yeah, that's incredible. So a couple of just of questions regarding society, we can say. So I think Alex will be able to ask these couple of questions. Alex, or I don't know if he has trouble with the connection, or
yes, Professor.
Yeah,
I'm here.
So, go ahead.
So if there were several warning and the pandemic has been developing from last year, why were we not prepared for this? The second one.
Yeah, that's a question that Sadia asked. Sorry, Sadia. But basically, why we were not prepared for that? That's a very good question. Yeah. And the second,
very good question.
Yeah.
There were programs to be prepared. I mean, I am managing a program that was instated by the federal government in 2015 to prepare hospitals and healthcare system to respond quickly to this type of infections, how to isolate and how to identify, isolate, and inform the authorities that this is that an special pathogen has come to the city. But I think putting together all this developed really fast. I think since in January everything went speeding up, and I think the problem with this pathogen is that it has been spreading very quickly to different, and it's very difficult to control. For example, with the SARS-1, the coronavirus one that Maite has said, people that were spreading the disease and they were contagious were those that were very severe. So you were able to isolate and to stop the spreading in the community very quickly. With this virus, the whole range of symptoms that people that get infected get is very difficult to isolate. So I think that has been making so difficult to respond to the pandemic, but I am sure that healthcare departments have been working intensively to cope with the disease, but unfortunately not quickly enough. And I mean, it has been developing for 2 months until when they came to the US, it was already only 1 month after it was declared. So it's not so long time in order to start working. And we are learning so many things for the virus. So because the problem is that it's completely new. So it's very difficult to respond to a disease that you don't know everything.
Mm-hmm. And Sadia, do you want to ask our last question, and then we will give a couple of minutes to last minute question. But Sadia, would you like to go ahead and ask the last?
Sure. So our last question was asked by Afnan Fariha, and the question is, as scientists, how do you feel when you know that the disease has killed a lot of people but being sold in laboratories?
This is, thank you very much, Sadia. This is a question that is being out there regarding to the conspiracy theory. I don't know if any of you would like to
Well,
talk about that. Maite, go ahead.
yeah, I mean, as a scientist, I believe in the real data and the data you can get from the research. And there is not data about that this virus could have done in a laboratory. So
I think that we had a conversation a couple of years ago regarding this, because Maite has been presenting this for another our college. So I remember that you were mentioning that the only moment that we realize that that virus is there, the amount of virus that there are in the planet is incredible, no? So we only notice them when they cause a disease, right? So
yes, exactly.
It's not. I am not going to say anything about theories that are not proven, but basically, based on a conversation that we had in the past, I think that it's very probable that the variety of virus out there allows for one of them to be a little bit more dangerous than the rest, right? Or more dangerous, right? I don't know.
Yes, exactly. There is so many viruses that we still don't know, and they are out there getting ready to infect us. That these kind of pandemics are gonna happen, and they happened before with the flu or with Ebola or with SARS. So
or HIV. HIV came from another animal, and it's a pandemic in the world, so it's not the first one. And the one thing that is important is if we as humans, we are interacting and we are destroying natural habitats where we get in contact with wild animals, we are going to have more chances that we get these diseases that usually should be kept in the wild habitats with those animals that they don't have disease. And but yeah, it depends on us how we treat the natural and high environments, so we become close to the planetary health, one health. Everybody, animals, humans, and environments, we are one, and we should be taking care of all of us.
I'm going to thank you very much for these questions. I'm just going to take a couple of short questions the students are asking in the chat. And we address many of them related to the infection, disease, and everything. And also, we are recording this session, so you will have the opportunity to watch this again later if you want. But so one of the questions that I think that it's in the air is if someone was tested positive for COVID-19, how soon before they can return back to work? That's a question from Kiara. Do you have any advice? That's also related to the question that I have of how long do you have to be in quarantine so you don't infect others, your family or your friends or people outside. And also the situation of getting back to work, we need to work, right? So we need to pay our bills, right? So
yeah, the instructions is what I said. A minimum of 7 days you have to be at home after the beginning of the symptoms, or 3 days after the fever resolved. So whichever is minimum. So maximum, the minimum, sorry.
Yes, okay.
So, and yeah, but if you have any questions, the 311 form from the city would be able to answer those questions specifically about that.
I think that we have addressed many of the questions that we have in the chat. But if you will have any specific questions, you can raise your hand in the Zoom, and I will address that. But in any case, or when our expert will address that. I would like to say thank you so much for your participation. It was such an amazing presentation that you did, both of you, and also all these questions. This is something, information and education is power, right? And yes, we need to be protected. Thank you very much, because you did an amazing job. And our students have a much more information regarding this, thanks to you today. So if you would like to say a closing remarks, or you have the microphone open for you. Thank you again. Maite?
Thank you all of you for listening. I hope this helped to understand much better what is going on. And if you have any kind of questions, or you want to email us regarding anything,
yes, I'm going to post at the end of the presentation your pictures with your email, so our students, they can expand the network of connection, and at the same time they will be able to address the questions directly with experts.
Yeah, we are here for them. So yeah.
Thank you very much, Eli, and thank you very much, Elizabeth Diago Navarro, and Dr. Elizabeth Diago Navarro, and Dr. Maite Sánchez Aparicio. Thank you so much. You are very good speakers and you did a very good job. Thank you so much.